Hi guys, in this video we're going to talk about gene cloning. We're going to define gene cloning and then look at the bacteria and plasmids within those bacteria that are used for gene cloning. Then we're going to look at the method and some of the uses in both a gene library and in protein production. Okay, so first to define gene cloning. Sometimes gene cloning is referred to as DNA cloning or molecular cloning. And what it is just that. It's cloning a section of DNA, not a whole organism, just a target section of DNA. And by cloning that or amplifying that DNA, making more copies of that DNA, usually occurs through a vector. And this vector is quite often bacteria and usually E. coli. So to understand the process of gene cloning, we need to know a little bit about bacteria and the plasmids that are inside bacteria. So bacteria have inside them two types of DNA. So bacteria are prokaryotic. They don't have a nucleus. They've got their DNA floating around inside them. One of these pieces of DNA is the chromosomal DNA, which is found in a large sort of tangled ring. While another piece of DNA that they have inside them is called plasmids, and these are small rings of DNA and generally code for non-essential or extra traits within a bacteria. So it's not necessary for these for a bacteria to have these plasmids in it. And an interesting thing about DNA is they reproduce through binary fission, which is mitosis, so they get exact copies of each other, but they're also capable of this thing called horizontal gene transfer. And in this case, they actually can transfer genes from one individual to another through plasmids. And this process is called transduction. So basically what they do is they rub up to each other, against each other, and the cell membranes touch and open up and allow these plasmids to pass from one to another. So this actually changes the traits and the DNA inside a bacteria, and therefore changes the traits that they express. Okay, so now let's have a look at the method, and the method is called recombinant DNA. So firstly, in this gene cloning, we need to work out what our target gene is. So to do this, they map the genome and they have a look through the genome of the donor, and they work out the target gene and where it starts and where it finishes, so they can cut it out. And to do the cutting, they use restriction enzymes called endonucleases. And these endonucleases are specific to a specific site on the DNA. So it might be that they search along the DNA for a sequence that's ATGC-ATT. And then they will go all the way along the DNA, and then they find that sequence, and then they cut the DNA at that sequence, leaving these sticky ends. So we've got these restriction sites. The endonucleases, so endo is within rather than the cutting at the ends, they're cutting inside the nuclease. So that's cutting at that restriction site. So this is the gene, the target gene inside, and this is what we call sticky ends on either side of the gene. They then use the same restriction enzyme on a bacterial plasmid, and we've mapped a whole heap of different plasmids, so they choose the one with the a similar restriction site and work out where that restriction site is and use the same restriction enzyme. And when I say the same, we've got a library of about 3,000 different restriction enzymes that cut at different sequences. So by using the same one, it opens that plasmid up, so it's going to have the same sticky ends or the same complementary bases exposed as that of our target DNA, the piece that we want to actually amplify. So we then get that target gene and we mix it with the plasmid, and some of the plasmids or some of those genes will attach to the plasmids and they'll bind together with the sticky ends of the plasmid. Some plasmids will just join back up, But yeah, some will bind together, and then we can use DNA ligase to seal up those sticky ends and seal that backbone, and now we have a recombinant plasmid. So this is a plasmid that, as well as its original DNA, also has that target gene that you wanted to have. Now, a lot of these plasmids that they use also have genes in them for antibiotic resistance, and some have markers on them as well that might make them a particular color, so that we can tell whether this process has worked and whether the plasmids that we've got are the recombinant ones or not. That modified plasmid is then introduced into bacteria, and then a heat shock or electric shock is used, and that basically opens up the pores, or kind of like pores, of a bacteria to allow those plasmids to slip inside. So what we're going to then have, and this process is called transfection, and what we're going to have then is some bacteria that have this recombinant plasmid inside them. Not all bacteria, but some of them. We then can take that modified bacteria and grow it on a nutrient plate, so like an agar plate. And because of the markers and the antibiotic resistance, we can use these traits to know whether the bacteria that we're growing has got that plasmid inside it, or whether it's just got the plasmid that's joined together, or whether that plasmid hasn't actually gone into the bacteria at all. So those we're able to use those to work out which ones have been successfully modified, and now we have our bacteria with recombinant DNA inside it. So it's got that gene that we want inside. We can use this recombinant DNA to produce what are called gene libraries. So instead of just cutting out the one target gene, what we can do is use a number of restriction enzymes to chop up the whole genome at various spots and put all those different fragments into different bacteria, and then put them into those bacteria cells, so that bacteria cell produces the protein that's associated with that gene. So this way we can actually map what proteins are made from which genes in the genome. What we can also do is then store these genes in a gene library, which has all the different genes that a particular organism has in its genome. And then if we want to use those genes for something like transgenics, where we want to, say, put a jellyfish gene into something, we can go to the gene library for the jellyfish, and it's a stack of petri dishes with bacteria growing on them, and we can choose the particular gene that we want and then isolate it out of that plate. Another way that we can use these recombinant DNA, or this gene cloning, after amplifying the gene, is by keeping that bacteria, so rather than just putting it on a plate in hibernation, keeping that bacteria and have it basically go through its life cycle, and as part of that life cycle it is going to express the gene. So this is a good way to produce a protein by taking the gene for the protein that we want, modifying a bacteria to produce that protein, and then we just have a culture of bacteria, and as part of their life they produce that protein. And this is how we currently produce insulin for the mass market. Previous to this technology, we had to harvest insulin from pigs, and pig insulin is very similar to human insulin, so it was something that we could use, but now we don't have to harvest this from pigs' livers. We can just have bacteria in a tank producing insulin for us. In this video, we have looked at gene cloning, or molecular cloning, the process of cloning a section of DNA. We've talked about bacteria and plasmids, plasmids being small rings of DNA found inside bacteria. We've talked about the method of gene cloning using bacteria as a vector, taking that DNA, the target gene, placing it into a plasmid, putting that plasmid into the bacterial vector, and then growing from there. We have talked about a gene library and how they are created and used, and we've talked about how bacteria can be used to produce proteins for us. Thanks for watching, guys. Peace out.